This is session 17. So session 18, the key and the worksheet are on the website, but I'm not going to do a recording of them. I'm getting permission from my boss tomorrow to do two final review sessions. The one for tomorrow will be a cumulative session, and I'll just go over the cumulative worksheet that's on the website with the key and talk about that. And then on Thursday, it will be the new portion. Unless those are other way. Maybe Tuesday's new and then Thursday's cumulative. And then during those times when I would have SI session, I will be up on the Zoom to be able to answer any questions for y'all or just kind of chit chat or go over anything you need to go over still. I am dyeing some of my hair blue. Dyeing some of my hair blue. I actually will put the video right here. But I thought it'd be fun because I just don't want to do anything, and so I like clean my house and knit the dishes, and I'm dyeing my hair blue in an effort to not do any work. But we're going to do this session because I have got to do other things, and I'm making the choice, so it's happening. Okay, so we're going to do this right meow. All right, so this is my wonderful screen and my grandmother and everything. Cool. So okay. All right. We're going to do this quickly and professionally, and it's going to be great because my hair's done soon. So we're going to do this quickly and have fun and learn a lot of things so that we can all go enjoy the rest of our Monday. Okay, session 17. So this is all about axon functions and different kinds of neurons and how they fire, more of the physiology of it. I feel like last couple sessions we did were about, we're about the names and the structures, and this will be more physiology heavy. Okay. All right, so the axon is a conducting region of the neuron, right? So where's my little doodad? This, right? The axon is the conducting region of the neuron. That's where our action potential, which is what this little wave is, runs down, right? So say we get, let's see, where's the thing that allows me to draw? Cool. So say we get an impulse right here from another neuron. It'll travel down these dendrites through the parakaryon or the soma, right? That's what this is. That's just, we'll look at that later. It's like on a graph. It would be like negative 70 millivolts, depolarized, action potentials up here, right? We have an axon potential that's fired, and then we go back down to negative 70 millivolts. Anyway, which is what this is. So we go down the axon, and then we transmit this down here to the axon terminals, which maybe will be at a muscle, an organ, a gland, an effector organ, anything like that, because we're just sending the signal, right? It's a conducting region. We're conducting electrical impulses, which tell us what to do, which tells us to blink, tells us to move our eyes, tells us to do anything we're supposed to do. That's how we communicate. Okay. And good. I'm gonna just leave this doodad up. Okay, so it generates a nerve impulse that travels along the axolemma, right? We said a sarcolemma was the membrane, the cell membrane of a muscle fiber. So the axolemma is the cell membrane of a neuron, okay? So it transmits along the axolemma to the axon terminal, right? To the end. This is an axon terminal, right? The terminal is where we secrete that neurotransmitter, right? If you remember in the muscle video, we saw that acetylcholine come out of the axon terminal. Okay, remember there were these little secretory vesicles, and inside of them there were the triangles, and that was acetylcholine, and those came out of here because we had an action potential. Sorry, this is challenging. An axon potential that came down this, which told these and the acetylcholine or whatever hormone it is to come out, right, and to signal whatever cell. They're all my drawings. Oh, that's easier. Okay. Okay, and what can it do? It can excite or inhibit a neuron, right? So it can either tell it to start or stop or continue or get hard or increase or decrease, okay? It communicates with many different neurons. It communicates with many different neurons at the same time, right? So that's why we have so many dendrites and we have so many axon terminals, because we can send one message down lots of axons, and it will transmit everywhere, right? So all of these, right, we send a message here and here and here and here and here and here, okay? Okay. So a myelin sheath, right? So they're composed of a myelin sheath, which is a white protein lipid structure, okay? So protein and lipids, so it has proteins and it has fat in it, and it's a structure substance, right? And its function is to protect and electrically insulate and increases the speed of the nerve impulse transmission. That's where our lipid comes in, okay? Our lipid increases the speed of the nerve transmission because it allows us to have these things called nodes of Ranvier and those myelin sheaths, which we'll look at in a minute, so do not be afraid. That allow us to have that electrical signal jump, okay? So it's composed of a myelin sheath, right, which protects and it increases the speed of transmission, right? We want to make sure, remember that in your nervous system, that's our fast acting response system. And so we want to make sure that we're doing it fast. So we want to make sure we're getting the message, right? So it's insulated and it's going to get there and that it happens quickly, okay? So there are fibers that are myelinated and there are fibers that are non-myelinated. Where do I want this? There are fibers that are myelinated and fibers that are non-myelinated. So myelinated pretty much just means they have this myelin sheath. They have this white protein lipid substance that protects and insulates and increases the speed. So if it's a myelinated fiber, right, it's surrounded by a long axon that has a myelin sheath. And so it's going to do these things. It's going to have protection, insulation, and it's going to have an increased speed of transmission. However, the non-myelinated fibers will not, okay? So these do not contain a sheath and they happen more slowly, right? Our electric impulse goes more slowly. Okay, so myelination is formed in Schwann cells, right? So if you don't remember what a Schwann cell is, go back to the last session or to, where is it? I want you to see this video, but also, way. But I want you to look at this video, okay? Divisions of the nervous system, and it's in that helpful exams for video, or for exam right now. Sorry, when you're applying to med school, you get a lot of med school ads. I find it interesting how like ads are targeted to everyone. Anyway, go watch like Jeopardy at like 9 a.m. and you'll see some very different ads. Anyway, this video, where's the frontal breakdown? Okay, it gives you all the divisions of the nervous system, and it talks about what kinds of cells and things comprise each part, right? So it's important, as well as this video where we talk about each kind of cell in each place, right? And so it's important to make sure that you can differentiate different kinds of cells and their function based on whether they're in the central or the peripheral nervous system, okay? So myelination is formed in Schwann cells, and its function is to wrap around the axons in a jelly roll fashion. Okay, so, which pretty much looks like, give me a neuron, come on. Okay, that doesn't want your myelination on it. Oh, there it is, there's one. Also, this guy is awesome, if I'm being honest. I watch him for a lot of like physics and MCAT stuff, and I think he's really good. So, okay, right, so these, right, these little blue pieces are the myelin sheaths. Those are those wrappings of the myelinations that are formed by Schwann cells, which allows us to have an electrically insulated and protecting fiber, and it also allows us to increase the speed of impulse, okay? So that's what these look like, okay? And so then they form these nodes of Ranvier, which is the thing I said earlier, right? They're gaps between the Schwann cells, right? Or those little bubble jelly roll things that acts as axon collaterals and can emerge and allow us to have a faster conjunction. So, right? So we get an axon, we get a thing, okay? And then the action potential, right? It's chilling, it's going down, right? And then it goes to here, right? But instead it goes like this. It jumps from here to here to here. It jumps from the parts that are insulated to the parts that are non-insulated, right? So it jumps here and here. So it goes faster, okay? Clear all my drawings, okay. Okay, so those are the nodes of Ranvier. Those are super, super important. All right, so myelinating sheaths in the central nervous system, right, are formed by oligodendrocytes, right, which are similar to Schwann cells, which might means that Schwann cells are in the peripheral nervous system and oligodendrocytes are in the central nervous system, right? So they're formed by oligodendrocytes, but not the whole cell. Okay, we don't wrap the whole cell, we just wrap that axon, right? So its function is to wrap axons. Okay, so we have white and we have gray matter when we're talking about the central nervous system, right, which is our brain and our spinal cord. So white matter is a dense collection of myelinated fibers and fiber tracts. So it makes sense that the white matter is going to be lots of myelinated fibers, right, lots of lipid and protein heavy wrapped fibers, right, which gives it that white character, okay? And then gray matter is mostly cell bodies, right, or non-myelinated fibers. Okay, so picture this. Whatever we're doing, fibers, axons, all that jazz is originally gray, okay, which is why the gray matter is non-myelinated. However, when we myelinate something, right, when we give it that axon wrapping, right, of the myelin sheath, it becomes white. So then it's white matter, okay? Remember that we're talking about the central nervous system, okay? Okay, so we have lots of classifications of neurons, which is where our function and our classification and our branching really comes in here. Okay, so we have the main classifications are structural and functional. Okay, so they'll classify them by their structure or by their function. Okay, so in our structure, we have multipolar, bipolar, and unipolar, right? And I'll show you these in a second. And then under unipolar, we have peripheral and proximal, okay? Functional, we have sensory, motor, and interneurons, right? Either it senses something, it transmits an impulse to tell us to do something, right? Like this, right? So I'm either touching the pan and it's hot, and then I'm getting a sensory neuron and it's telling my brain like, this is hot, don't touch it. Or I'm having a motor neuron, right, that tells me to move my hand, right? And then we have these interneurons, which we'll look at in a second, which are the connection between them. Okay, so we have sensory that are going right to my brain, and then we have motor that are going out, okay? And then we have neurons that come between them. I'll show you. Okay, so. All right, so this is unipolar. Let's see.
This neuron, as the name suggests, only has one process.
Okay, so this is our important piece, right? It may have this long axon and it has dendrites on this side and dendrites on this side, but it only has one process, okay?
Referring to one. And this type of neuron is not truly found in humans. It's only found in invertebrates. It says would extend inside these drawn this with an at the top. So
okay, so then we have bipolar that has two ends. It has two axon pieces, okay? So we could go either direction, right? With that unipolar one, we can only go from here out, okay? And usually it will go here, and then it will go down or it will go up, right? But with this bipolar, right, we can come from here and go to here. We can come from here and go to here. And then we have multipolar, where we can go lots and lots of different places, okay? We can come from here and go to here. We can come from here, right? There are lots of more tracks, okay? So that's where our classifications of these come in. There's also a John Green video on this, and that's very good as well. I think it's in here. Okay, so I recommend that you listen to him talk about these to understand more of what they do, because usually on the exam they'll try and make you differentiate between these, and they'll give you like a scenario, and then they'll ask you which kind of neuron would do this part. Okay, so then we have functional, sensory, and motor and interneurons. Okay. Okay, cool. So also in this video, that's dope. Okay. Right, so we have sensory neurons, which are known as afferent neurons, right? They transmit impulses from the sensory receptor to the central nervous system, right? Okay, so so there's something on our leg, so that you see this little firing, it's going up towards the nervous system, okay? We're sensing something, right? Usually these are unipolar. I don't think you actually need to know that. And then we have motor neurons, right? So you see how it's radiating out from the central nervous system and it's going down to wherever we need it to be, so that it moves from the central nervous system, right? It's efferent, so it's exiting the central nervous system to the rest of the body, and these are usually multipolar. And then we have these interneurons. These are super cool, and I think they're really important. So these are the association neurons. So these are the ones that move between the sensory and the motor. Is it going to give me more? Okay. So these are in our spinal cord and in our brain. So they connect the part of us sensing to the part of us moving, and they're just the neurons that connect between. So I like to draw them like this. So if this is our, let's go back. So if this is our hand, because it's really pretty, I don't want to talk about the hand being really ugly because this is where we are in my life. Okay. Awesome. We get stung by a bee or something. Let's try and draw a bee. Oh, how about it? Boom. That's my bee. Cool. We get stung by a bee and it hurts, okay? So then we have a sensory neuron that, right, maybe it's like literally right here, and this neuron tells us that this hurts, right? So this is our sensory neuron, right? So it's an afferent neuron because it's going towards the central nervous system, okay? And then we have an interneuron, right? So this is our spinal cord, right here. And so we have a neuron that connects from here to the spinal cord to transmit this to the spinal cord. And then finally, right, the information comes back down, right? And we have a different neuron, right? Maybe this is our motor neuron. Okay, and then this is efferent because it's exiting the central nervous system, and it goes out and it tells us to move our arm or tells us to rub our arm that it hurts. So usually you'll have a motor neuron, right? The interneuron connects between the two, and then you'll have a sensory neuron because that's where we start, because we need the sensation to tell us what's up, right? So we go this way, right? And this is our central nervous system in here, okay? Right, we sense, we go to this, we interpret, and then we go out. I hope that makes sense. Okay. All right, so we have structural classification. So the multipolar has 3 processes. It has 1 axon and other dendrites, okay? A bipolar has 2 processes, right? It goes like this. It has 1 axon and 1 dendrite. And then unipolar has that 1 T-like process with 2 axons. Remember, it's got the like the head in the middle. Right, it's like this. And it's got jazz on the end. Okay, and then the bipolar is the one that looks like this. Right, but the difference here is we have this connecting piece between the uni, but the bipolar is connected directly to the axon, and then you know in a multipolar ones, that's the one we always see everywhere. Okay, so we have some differentiation between unipolar. We have peripheral and proximal. So if you remember what proximal and distal mean from lab, you know that the proximal one is going to be proximal, right? It's going to be closer towards that joint or towards the midline, right? So if you don't know what proximal and distal is, look them up. However, here's an example. My wrist is proximal to my elbow. Okay, so it's closer to my elbow. Proximal is closer, distal is farther away. Okay, so my fingers are distal, right, because they're really far away. However, my shoulder is proximal. Go look it up if you don't know. I find that that will be easier. Okay, so peripheral, right, is going to be our distal. It's going to be our sensing, right? So if we're saying fingers are distal, right, these are a lot of our sensors, okay? And then proximal is our central. It enters the central nervous system, right? We're getting closer and closer to the midline. All right, so our functional classifications, we have sensory, right, where they send the sensation to the central nervous system. We have motor, where they carry the impulse from the central nervous system to the effector or the excitor, right, which pretty much just means my brain knows that I need to move my arm, and it's sending that impulse from my central nervous system down to my hand to tell me to move my arm. And then we have interneurons, also called association neurons, that are between the motor and the sensory, right? They signal through the central nervous system, which is what we talked about seconds ago. Right, so literally, this looks exactly like your muscle fibers. Do you remember? Right, perineurium, epineurium, endoneurium. It's the same stuff just with neurons. So if you had a really good time with the structure of muscle fibers, you're gonna have a great time with this structure. It's the same, right? And you know where epi is, you know where peri, and you know where endo is, okay? Okay, so nerves are just cord-like bundles of axons that conduct sensory and motor impulses, which is what we've been talking about the whole time. It's a cord that conducts sensory and motor impulses, right? It conducts that our arm hurts, it conducts that we need to move our arm, okay? So our smallest, right, is our endoneurium, right? It encloses the axon and Schwann cells of individual neurons. So it's the smallest covering. And then we have the perineurium, which bind fibers into fascicles, and it covers each fascicle, right? So it covers each whole fascicle, right, which is this. Okay, oops. And then we have our epineurium, which encloses the whole nerve, right? It encloses lots of fascicles and it's our entire nerve, right? We also classify these by the direction they transmit, right? Efferent and afferent. We talked about this. This is so important. They ask you this on the test all the time. They want to make sure that you know what you're talking about and you can distinguish between these, okay? So my trick is efferent is exiting the central nervous system. I can type that in here. So efferent is exiting the central nervous system, right? So central nervous system to somewhere else, okay? Where our afferent, it affects what we do. It affects what we do, right? So our sensation goes towards the central nervous system. However, you only have to remember one of these and then you know what the other one does. So I like to say that the efferent exits the central nervous system. So it goes from your brain somewhere else, okay? Okay, so, right, so then we have mixed nerves which do both, right? So they go efferent and afferent, okay? All right, so now we're gonna talk about the physiology of a nerve impulse. Okay, so bear with me. A lot of this is very, very similar to muscles. So if you understood the muscle contraction and all that, you're really going to understand this. Do not be afraid if you didn't. It's okay. You're probably going to have a couple light bulb moments now that you're doing it again, and you might get it better. So it's okay. So neurons have a resting membrane potential, just like muscle fibers. Remember, we said that negative 70 millivolts. Unlike most, neurons can rapidly change resting membrane to potential, right? Remember, we talked about how that happens with ion movement across the membrane. We'll look at that soon. That ion movement allows that resting membrane potential to change, okay? Remember, everything about anatomy and our bodies is about homeostatic balances, so we want to make sure that we're in the right area, and so if we're changing something, something is going to happen, okay? Neurons are highly excitable, we know that, okay? And they measure things in voltage, which is just a measure of potential energy, right? If we have a lot of negative charge over here and a lot of positive charge over here, we have a really high voltage because we have a lot of potential energy because there's two attracting forces really close together. And so our voltage is we have a really high potential energy because these are going to want to do this, okay? It's just the difference in potential energy across the distance is voltage. Okay, so it's called a potential or potential difference, okay? The charge difference across the plasma membrane results in a potential, right? It's just potential energy. We're just talking about neurons, or we're just talking about different, we're just talking about electrical impulses and the potential to do work or move things or fire an action potential, right? Okay, so the role, I don't want to, can you just stop now? Okay, so the role of membrane ion channels, right? We have large proteins that are selective for our membrane ion channels, right? Just like in our muscle, just like in any other cell membrane, is we have selective membrane proteins that allow things in and out and don't allow other things in and out, right? So we have potassium ion channels that allow potassium to pass through, and we have lots of different kinds of channels, okay? So let's see. We're going to talk about this and then I'll show you the video. It will make more sense. Please bear with me. Okay, so we have types of ion channels, right? Just like when we talked about ion channels the last time, right? We have leakage that are non-gated, right? They're always open, they're always leaking, okay? We have gated, there are proteins that change shape to open and close, right? This has a lot to do with the sodium-potassium pump, which I'll show you later. And then we have three types of gated channels. We have chemically gated channels. Remember we talked about those with muscles. We have chemically gated ion channels, right? That bind ligand gated channels. A ligand is just a protein. It's just a thing that binds that allows it to move, okay? And it only binds with specific chemicals. Remember we talked about how specific proteins and specific ions and channels and things have very specific lock and key mechanisms that allow certain things to go through and not other things, right? So examples are acetylcholine or I don't know what that says. I think it means, oh, I know what it says. Acetylcholine or neurotransmitters. It just says neurotransmitters, okay? We also have voltage-gated ion channels, which we've heard of, right, which open and close due to membrane potential change, right? When our ions move and we have a change in membrane potential, and then our voltage-gated ion channels will open and close depending on our voltage, okay? Then we have mechanically gated ion channels, which open and close due to physical deformation. We will look at, right? So if something changes, remember we said structure equals function. So if the structure changes, the function changes, and it's going to do something else. Okay. So again, just like in muscles, we have an electrochemical gradient. Ooh, don't want me. Where ion flow creates an electrical chemical current and voltage across the membrane. Electrochemical gradient, right? The membrane has a gradient, and the gradient is from ion flow, which creates an electrical current, okay? Let's go back to wherever we were. No, it's not. Nope. Nope. Okay, cool. So the axon hillock, do you remember, is this little cone shape right before the axon, right? So look, these are voltage gated ion channels, okay? And they allow sodium and potassium to move, right? So we see positive and negative charges everywhere. This is what we're talking about when we're talking about that electrochemical gradient. Ignore this. Okay, actually we'll watch. Okay, so we have voltage gated ion channels. We have ligand-gated ion channels. Okay, actually, let's just watch this for a second. I will put this video. Okay, so this video is for med school, so it's going to go too much for you, but I think it's important to see what we're talking about. So let's watch this for a second. Right, so we're repolarizing. Okay. This is our neuron, right? It's multipolar. So we're looking at all these positive charges, right? Which is changing our membrane potential positive charges, right? Which means we're getting impulses from these dendrites, okay? Which is changing our membrane potential, which finds us at a weird membrane potential, right? That we don't want to be at. And this is important, right? Our threshold potential, right? Remember in muscle fibers, our threshold potential was if we reach this, we will have an action potential generation, right? It's all or none principle. It's either happening or it's not happening. And if we reach that threshold, it's going to happen, right? We're firing, okay? Right, more positive, it's getting bigger, it's getting more important. And then these voltage gated ion channels, because of this, are going to allow, right, so we have all that positive stuff, right? Right, it's going to allow sodium to run in, okay, increasing our threshold, right? We're getting higher and higher. We've reached the threshold, so it's happening. We're just getting there, right? So we're going in. Right, so now we're going to have an action potential that occurs. Okay, so literally, this is that graph we were talking about. So our electrical chemical gradient is when our voltage and those positive and negatives that we're looking at right here are so great or so different. Why can't I find it? Are so great or so different that we cause an Axon potential. So this little graph is kind of just showing us what happens, right? So negative 70 millivolts, maybe that's where we're resting or chilling, okay? And then sodium increases. Don't worry about that. Okay, we're going up, right? Our voltage is going up, and then we reach a threshold. Okay, right, so we're having more potassium, right? So this is our threshold, and then we fire our action potential. We'll talk about refractory periods. I feel like we did kind of talk about them. We talked about absolute and, or an absolute refractory period is when we can't do anything else. We have to make sure that we come down and get that membrane potential back to enough normal that we could move those charges again to refire. And then we have relative refractory period, which is where we're kind of close to back to normal, but we're not quite there yet, but we could. It's like, so absolute refractory period is like the minute after you take a test and you're like, my brain is dead. I cannot do anything else. I need to get some spoons, which is what we call me time in my house, right? I need some spoons or some, like, my brain needs to chill and watch something dumb and recharge, right? So we can't do anything. I'm in absolute refractory period. I need rest. Relative refractory period is like, on episode 3, okay, my brain is like kind of back. I'm still like kind of tired, but I have other homework to do and I probably need to do it. And now I could do it. Okay. That's how I like to explain it. Right. And then at some point we come back to resting membrane potential. Okay. Right. So these are our nodes of Ranvier, right, which is between these Schwann cells from oligodendrocytes. You see these little myelin sheets. Is it going to show me? So, right, so positive. So we're about to reach action potential, right? We reach the threshold and then we're going to fire and they're jumping. You see they jump around them. And then, right, these are the neurotransmitters of the acetylcholine. Right, so this is our action potential and it fires and it jumps between them, which is why it makes it faster. Okay. I know that was a lot. Electrochemical gradient is that we're talking about the difference between both sides of the axonema. Okay, so our resting membrane potential, where's my stupid little thing? Our resting membrane potential is negative 70 millivolts, okay? Right, our sodium potassium pump stabilizes our resting membrane potential and maintains the concentration gradient. So it's literally this little pump and it has studs for both on there. Right, so it pretty much looks like, why is this being mad at me? I'm sorry, hold on. My computer is angry. So it's literally this little protein. Okay, that's not good. Can I stop being there? Can I go away? Okay. Oh, it's freaking out. Okay, so it's literally this little protein, right? And on one side it has places for sodium, and on the other side it has places for potassium. It looks much prettier. Okay. So our sodium potassium pump will let some sodium in, right? And then it will flip and it will be this way. And it'll let the sodium out. And then it'll flip back, right? And then it will grab some potassium on its way out. It'll grab some potassium on its way out. And then it'll flip and it'll let the potassium out. So pretty much the point of me saying that is it just maintains the concentration gradient by making sure that enough ions are moving so that we stay at resting membrane potential, okay? So the sodium potassium pump, we have 3 sodiums for every 2 potassiums. So if we let out 3 sodiums, we're gonna bring back 2 potassiums, right? So these are our ratios, okay? So the membrane potential changes, right? So the concentration of ions across the membrane changes and the membrane permeability of ions changes, okay? So that's how we get a potential. Right, so during a potential, our concentration of ions change, right? We're not at normal anymore, and our permeability changes, right? So we allow more sodium in. Do you remember that? Right, so a change produces 2 types of signals. We have a graded signal, a graded potential, and an action potential, right? A graded potential is when we're slowly creeping up, okay? It's an incoming signal that comes from short distances, right? But an axon potential, right? So a graded potential goes it goes a little ways, right? But an action potential travels the distance of an axon. It's a huge distance for that charge, relatively, okay? So then we have depolarization and hyperpolarization, which is just, okay. So this is our beautiful little axon, right? So remember we have threshold, right? We have threshold right here, right? And then we fire an action potential, right? Right, so we're depolarizing right here. This is all depolarization. Okay, we're getting depolarized. We're getting more towards positive, which I know, right? So technically if 0 was like way up here, right? We're getting more positive. We're depolarizing, right? And then we go back down. We get closer towards our resting membrane potential of -65 apparently in this case, right? But ours is -70, right? We get closer, and this is repolarizing. So ugly, I'm so sorry. Repolarizing, right? Because we're getting closer. And then it does this thing and it dips. Okay, it dips. So this dip, right? We've gone past this -65 right here, right? This is hyperpolarization. We've gone too far down, right? So this is hyperpolarization where we've gone too polar, right? We've gone too negative. And then at some point we'll come back up to that -65, -70, right? So these are our hyperpolarization and the votes, okay? Our hyperpolarization and our repolarization. Right, so depolarization, we change the membrane potential to become less negative. That's when we're going up, right? And then hyperpolarization is we're too negative and we're decreasing our amount of impulses, okay? So our gradient potentials are short-lived, they're localized, right? And they have stronger stimulus and more voltage changes, right? So the triggered stimulus equals an opening of gated ion channels, right? They're named for their location and their function, right? We have a generator potential and we have a postsynaptic potential, right? So the generator potential is also known as a receptor potential, and these have to do with sensory neurons, okay? Right, so generator, think of a generator starts something, it starts a process, it starts the AC, it starts the gas, so it's a sensory neuron. It has to do with us receiving information or a receptory potential. And then postsynaptic, right, meaning after the synapse, right, is a neuron graded potential, okay? So it's afterwards. All right, once an ion gated channel opens, the depolarization spreads. So remember how those little sodium channels were opening and allowing sodium to come in? So those gated ion channels were opened and allows the depolarization to happen, right? Which means we're getting more positive on the inside. And so we're going up and up and up and up and becoming less negative, right? Closer to 0, okay? And so the current flow dissipates and quickly and decays, right? So they're signals that are only meant for short distances, okay? So these graded potentials don't go really far. They're meant for smaller distances, okay? So then we have action potentials, right? This is the principal way or the most common way that neurons send signals. It's a neural communication where they occur at muscle cells of axons of neurons, right? So this is us literally talking to muscles. It's literally talking about an action potential to a muscle, right? So we're sending an impulse to or from a muscle, right? Action potentials do not decay over time, right? They maintain their strength from our brain all the way to our muscle. However, graded potentials do decay over time. So if they had to go a really long distance, whatever we would say up here, how strong it would not be nearly as strong as when we got down here as a graded potential. So nerve impulse is also known as an action potential. How are we doing? We can do this. Ready? 2 more pages. We can do it. Happy birthday. So I wonder how my hair's doing. Well, it's supposed to be blue. It's blue. I don't know. We'll find out at the end of the video. Right? The longer it sits, better. I don't know. I don't do my hair. I'm not really good at being a girl. So, all right. Steps to producing an action potential. So we have our resting state first, right? We're at that -70 millivolts. Everything's peachy keen. We're chilling. Okay. And then we go to depolarization, right? We get closer to 0. Okay. And then we repolarize after we've done that action potential, after we've generated that and fired whatever we needed to. and then we repolarize, we get closer back to our resting membrane potential, right? And then we talked about hyperpolarization, right? Which is when we go too deep, right? We're too far, and then we come back up to our resting membrane potential. So these are the nitty-gritty facts here. The resting potential, right, our leakage channels. I'm not going to explain all of this to you. I just don't. You need to know during what state, what is closed, what is open, and what comes in and out, right? So during depolarization, we have an influx of sodium, right? And it allows us to have more polarization, and therefore we get higher, okay? And then we have repolarization where sodium, or where potassium comes in, right? Sodium is no longer active because it was active during depolarization, so it's not going to be active during repolarization, okay? What is it called when the membrane returns to resting membrane potential? Repolarization, right? We're going back down to find back to our base, okay? I would just need to know what channels are open when, okay? Right, so the hyperpolarization is dipping below resting voltage. Okay, so do all action potentials produce depolarization? No, remember, they have to reach that threshold before they can fire and they can depolarize, okay? So if we don't reach the threshold, we don't depolarize, we don't produce an action potential, okay? An axon will fire when the depolarization threshold is reached, right? Which is what we just said. So on the all or none principle is talking about how an axon either happen or an action potential either happens completely or doesn't at all. Just like in muscles, there's no half contraction, there's no half firing. We either do it or we don't do it. Okay, no wishy-washiness. Okay, and then propagation allows axon potentials to be transmitted from origins down the entire length of the axon. So we're propagating it from one place all the way down to the other. We want to make sure that it gets all the way down, so we're propagating it through the entire axon. Okay. Once initiated, an axon potential is self-propagating. So it will occur forward always. Once we hit the threshold and it's time to happen, it's going to go. We're going to go the right direction. Okay, right? All right, so now we talked about this refractory period, right? The absolute and the relative. All right, so the refractory period is when we can't trigger another action potential. We can't fire again. We're not ready. We need rest, okay? And the sodium channels are open. We have two types. We have the absolute and that relative. Remember, absolute is when absolute is when we're right after the test. We can't do any more work. We need a rest. Our membrane potential, our ion gradient is not at a place where we could even have a contraction, where we could have an action potential or a fire. Nothing is happening. We need to chill. We need space, okay? We need to rest. However, our relative is our episode 3, and we're kind of chilling. We don't really want to do work because we're not quite back to where we need to be. But if we needed to, we could do something, right? So the absolute comes first and then the relative, right? Because you're tired and then you get less tired, okay? And so that's when we could actually fire an action potential. I think that's what you need to know. This is more information if you'd like it, okay? All right, action potentials only occur in axons, right? They only fire down axons, right? They have to get through that, that little triangle, which is called the axon hillock. It has to go through that and it has to be transmitted down the axon. An axon potential conduction is called a velocity. You can actually measure the velocity that an axon fires down, right? How fast it goes. Okay, so axon potential propagation, right? How fast it goes down the axon depends on a couple of things. It depends on diameter. So if there's a bigger diameter, there's less resistance and there's more space for us to move, and therefore a faster impulse. So think about a highway. So if you have a bigger highway, you're going to have less resistance and you're going to have faster traffic, right? There's not going to be as much traffic. It's not going to be as backed up, okay? Bigger road, less traffic. And then we talk about our degree of myelination, right? We talked about how they increase the speed of the nerve impulse. So how myelinated it is tells us a lot about how fast this is going to go. So there are two types. There's continuous conduction where it's slow and it occurs in a non-myelinated axon. So we're continuously chilling. We're kind of Priusing it down the road, right? We're not going really fast, but it's happening, right? This occurs on non-myelinated fibers because we don't have that nodes of Ranvier to jump. And then we have salutatory or saltatory, I don't know if I can say it. right? Which occurs in myelinated axons, and it occurs 30 times faster. It's literally so insanely faster. So myelin sheaths insulate and protect the leakage of charge, right? So it's keeping it localized, right? And then the myelin sheath gaps allow us to jump from gap to gap, right? We talked about, looks like this. It's so not happy with me. So if we have this beautiful axon that I'm drawing, and then we have, these are our myelin sheaths, okay? And then this is our axon potential. So we're coming down and we see this gap and we go, oh, we jump to here. Sorry, let's go back. Let's do that again. Action potential, jump, right? So this is our node of Ranvier right here, okay? And then we can jump again, right? So this allows us to jump from gap to gap, and it happens so much faster, right? 30 times faster. Oops. Okay. So here is our, right? So we have our nervous system. We have central, which is brain and spinal cord. And then we have peripheral, which has somatic, right? Which is our moving, it's our body, it's voluntary. And then we have autonomic, things that happen automatically. We have sympathetic and parasympathetic. Sympathetic is fight or flight. I say that your body is sympathetic, that you're gonna die, so it's gonna try and help you out. And then we have parasympathetic, which is that rest and digest, right? And so then this tells you which kinds of cells are in where. Peripheral nervous system, central nervous system, what they're called, and what they do. Okay. I am tired. And I hope that helped a little. And I will record test preps and I will let you know everything. Let's go. I mean, honestly, my hair isn't gonna look right, so I'll show it to you next time after I wash it out. But see you all later tomorrow for session, hopefully. Please watch these videos. They're really good. The sound is probably super important, and it will kind of show you a lot of what's happening, which I think is really important because the visual is more important, or is more helpful than me just saying words at you. So let me know if you need anything. Okay, bye.